For the n-Lorem Foundation, N of 1 doesn't mean alone because every patient matters. Hope for nano-rare patients and their families has never been more within reach. Visit n-Lorem.org today to donate and make hope possible.
Join us at the 2024 NanoRare Patient Colloquium hosted by Biogen on October 30th through 31st in Cambridge, Massachusetts. At this event, n-Lorem will review the Foundation's progress, present case studies, and host discussion panels with members of the NanoRare community. You'll also have the opportunity to connect with supporters, physicians, advocates, and, of course, patients and their families. We hope to see you there. Visit n-Lorem.org to register today.
This episode of the n-Lorem Patient Empowerment Program podcast is proudly sponsored by Hongene Biotech. With more than 26 years of experience in the nucleic acid industry, Hongene is a specialized CDMO and raw material supplier committed to providing high-quality and high-value products and services with the best-in-class lead times.
Hello, everyone, and welcome to the n-Lorem podcast series, the only podcast series devoted exclusively to the needs of nanorare patients and their families. I'm Stan Crook. I'm the founder and CEO of n-Lorem, and I'm your host for the n-Lorem podcast series. And today, as our special guest, we have Dr. David Butler from Hongene joining us. David, welcome.
Thank you very much, Stan.
It's good to have you here. And as a couple of grizzled oligonucleotide veterans, it's always good to get together and talk a bit.
Absolutely.
I think you're more grizzled than I am. Just to say it. Of course, it's hard to tell when you get to be my age what's more grizzled than anything else, right? But anyway, enough of that.
Yeah. I certainly have a grizzled look about me. So.
It's that Scottish background, it just, leads to a lot of gristle, as they say. Anyway, welcome. If that didn't make you feel welcome, I don't know what will. So why don't we begin by just introducing folks to Hongene, because I suspect most of our participants really don't know much about Hongene, and then we can go from there.
Okay, certainly. So Hongene is a manufacturer of RNA building blocks and oligo CDMO services and RNA CDMO services. It's been around now for about 25 years, based in China, but it's very much a global operation now. And yeah, so it's founded, the founder of the company is a gentleman called Wei Yang, and he founded the company based on a technology, chemoenzymatic methods to make nucleoside building blocks. And those nucleoside building blocks are the key building blocks for manufacturing phosphoramidites, which are, of course, the starting materials for oligonucleotide drugs. And we've been making phosphoramidites now since about 2006 and scaling them up. And we've forged some partnerships with companies, early companies in the space, including Ionis, n-Lorem, companies that are well recognized today. And we really partnered with those companies early on to help them to realize the cost-effective manufacturing for those starting materials. We've since applied that model to raw materials for mRNA manufacture as well. The IVT reagents such as NTPs, cap analogs, and enzymes, so that all of the key ingredients for manufacturing mRNA. And we applied a similar model where we partnered with companies early on, companies who are now very well recognized today, and help them on their journey to the clinic and eventually to commercial phase of development. So we've been doing that since about 2012 on the mRNA side of things. And then since about 2019, we've been getting into the oligo CDMO business, and that's maybe not quite as well known. I think we are an established player, I would say, in the raw material side of things, but for CDMO services, we're really more of a recent entrant into that arena. But we're making good progress, and manufacturing oligonucleotides is very exciting for us, and it's very exciting to be kind of a part of that entire value chain, going all the way from nucleoside building blocks through to oligonucleotide drugs and mRNA drugs. The majority of the operations are based in Shanghai. But we do have sites throughout the world now, and in the US on the West Coast, pretty close to you, we have a small site in Union City, in California. There's about 40 people there, and we do a combination of innovation projects and custom synthesis there. And on the East Coast, we're growing our US presence, and we recently set up offices on the East Coast, and we're building a small team out here, and that's partly my responsibility as well. And in Europe, we have a site in Hamburg where we have sales and distribution from that site, and we'll be expanding our manufacturing operations into Europe by the end of 2027. With a site in Hungary that's earmarked for opening in 2027, and then we have various sales and BD offices dotted throughout the world as well. So we're about 1,600 people, so not really small, quite a reasonable size, and really, so our value proposition to the field is that we're fully vertically integrated, so we can supply anything from the basic building blocks, nucleosides, all the way through to fill-finish drug. And we can do that in a way that's cost-effective, and we pride ourselves on providing material that's of the highest quality possible, which, of course, for oligonucleotides is very important. The starting materials have to be very pure, otherwise, as you know, Stan, the impurities can snowball. So a small amount of impurity in one starting material can lead to quite a lot of impurity in the final drug because of all of the iterative couplings involved in the manufacture. So that's Hongene in a nutshell.
Well, thank you for that. And so I'm interested and attracted to Hongene because it is a true entrepreneurial endeavor, like Ionis, like n-Lorem, like many of the companies that many of our listeners know very well. And it was one of the earliest biologically or medically focused startups out of China, as I recall. So it's been interesting to watch it grow and help the field grow and prosper while it also grew and prospered. And put that in context for people. One of the first things that people said to me in 1989, I have a list of dumb things people said to me in 1989, was that we would never be able to make oligonucleotides on scale. And having been responsible for scaling up small molecules and a lot of other things over my career, I felt that actually it would be a straightforward process to do that. And one of the challenges in manufacturing any drug is that you have to be in control of the supply chain so that every step in the way, not only is there a vendor available or you do it, and that vendor has to be high quality. And in the early days, we got building blocks for building oligonucleotides from salmon sperm, if you can imagine that. You must remember that, David. and that was one of the more terrifying ideas to me, was if we ran out of salmon, we might run out of opportunity. And so one of the things that Hongene, and among others, did was find chemically sensible paths to making the building blocks so that we're no longer dependent on a natural phenomenon that can change as the climate changes and so on. And as Hongene succeeded in that and providing the materials, then it moved on to do what made sense, which is become a manufacturer and formulator of oligonucleotide products. And so it's been it's been very I think synergistic. The field grew and Hongene grew, and they were sort of hand in glove to the whole process. And of course, you've been involved in a wide range of different activities in this space. So you've had pretty much the same opportunity I've had over the years to watch all that happen, and how actually remarkable it is that we are where we are today compared to where we started in 1989.
Yeah, no, it's amazing how far we've come. And yeah, my friend Yogesh, who you know, likes to tell the story about the salmon milt. And yeah, it's you can only imagine the impact that would have today if we had to isolate from salmon milt.
Both cost, it was the cheapest way to get it at the time, but it would today be very costly compared to the way we do it.
Yeah,
You
I
know,
mean,
we brought costs of oligonucleotide manufacturing down from billions of dollars a gram to really very affordable costs, and a whole set of inventions had to happen to make that happen, and Hongene played a role in a lot of that. So we're happy to have Hongene as a long-term partner in this endeavor. And I also want to mention, and this is very important, that Hongene is a strong supporter of n-Lorem. And I'm sure our listeners would be interested in how that came about at Hongene, and why that commitment is high.
yeah, absolutely. Well, so I think it begins with our mission, right? I think our mission has some crossover with n-Lorem's mission as well. So our mission is that we're continuously innovating to make RNA medicines accessible and affordable for patients globally. And there's nothing in that mission statement that's exclusive about, like prevalent diseases. So we see this as a call to arms for us to provide medicines for patients, regardless of how regardless of their economic means, but also there's treating patients on the asymptote of human disease, genetic disease, and that's all the way down to patients with individualized genetic mutations. And so that's how we find ourselves in this space and committed to this space. And we're, very, enthusiastic and happy to be committed and, partnering with n-Lorem, as we do also with the N of One Collaborative. So yeah, we're also sort of supporting that organization. So that's how we find ourselves, in this supporting role for the n-Lorem.
Well, we appreciate it, and it's an important role, because without the building blocks, we can't make oligos, and without oligos, we can't treat anybody. So having Hongene as a strong supporter is important to our patients, and I think that's important for folks involved in n-Lorem's mission to understand. Again, there are 1,000 things that go on in the back rooms of organizations to make that product that ultimately helps the patient that we treat, and every step in the way has to be high quality, and every organization like Hongene contributes to what we're able to do in meaningful ways. So we appreciate it.
Absolutely. Yeah. It's really a community, right? There's, I think many companies and individuals, people are coming together to do this because I think it's also, in addition to fitting with mission statements and all that kind of stuff, it's also the right thing to do. And we all feel empowered by that, I think, and really want to help n-Lorem to succeed.
Well, we're off to a great start, and certainly Hongene deserves credit for part of that good start. Of course, you're a chemist by training. I guess we should begin there, right? And
That's right.
your first experience in this space was at n-Lorem, if I understand it correctly, right?
That's correct. Yeah, I came to the US at the end of 2001 and worked at a CRO for a few years, but then transitioned to n-Lorem during 2007. And I was working in the research organization that was headed by Mano for 4 years, whom, of course, he's a Ionis veteran as well as an n-Lorem veteran, and well known and regarded throughout the field. So I worked as part of that team for 4 years, working on all sorts of interesting chemistries. Not the least of which I think was the lipid components of LNPs, which we developed into a drug, of course, patisiran. And we also invented the first biodegradable lipids, right? Which we thought we wouldn't need those anymore, and we kind of put those to one side back in 2011 because GalNAc was here, and GalNAc had ostensibly solved delivery for liver. And so we didn't think anything more about those lipids, but of course they made a big splash with the COVID vaccines, and they found a really nice home for delivery of mRNA and now gene editing drugs, and really they're having a renaissance, a major renaissance. And so I was there for 4 years, and then moved to Wave Life Sciences, where I was fortunate to develop stereo-defined oligonucleotides. I was involved in the drug discovery and development organizations there, and moved on to Alterna. And I don't know if you're familiar with Alterna, but
It's a tRNA company, right?
that's right, yeah. So it's similar in some ways.
I think tRNA is an overlooked RNA opportunity, that's for sure. And a good friend of mine, Sid Heck, has been in the business for a long time making atypical tRNAs that will incorporate amino acids. For our listeners, tRNAs are part of the process that leads to the synthesis of proteins. And you have a good many of them, one for each amino acid, just essentially. And there are opportunities to modify those therapeutically. So it's again, I think one of the things that you said, David, that I want to emphasize is the power of basic inventions. That when we make basic inventions like we did at Ionis and n-Lorem and so on, you never really know fully where the biggest impact is going to come. And it turns out LNP, which was eclipsed by something better to deliver siRNAs to the liver, or GalNAc, which, as Mano reminds me frequently, he did at Ionis first. And then, and of course, Martin Meyer was there too when you were there, right?
He was, yeah.
A couple of good chemists from Ionis that really, I think, helped n-Lorem a lot.
Yes, that's right.
The power of basic inventions is the basis of the modern world, and it's the basis of modern health. And so you do these things, and sometimes it leads to immediate giant success, and sometimes it has to percolate a while before an opportunity finds it that makes it a big success. And LNP is a great example of that, I think.
Yeah, I think you're exactly right. There's these technologies as well. They tend to kind of simmer a little bit in the background, and then yeah, all of a sudden, 5 or 10 years later, there's a new application for them. And so the advancements and the inventions that are somehow not lost. I think we're standing on the shoulders of the giants that came before us in many ways with respect to that. That type of thing. And yeah, I've seen that a lot. You know, I sometimes joke in meetings when we're discussing a new ligand or something like that, and maybe we're in a meeting with somebody that's kind of new to the space. I was looking at him like, hey, we were trying this 15 years ago at n-Lorem, and maybe in a different context, and things weren't working quite the same at the time, but now in a different context, maybe it's for a different modality, or there's improved chemistry on the oligonucleotide backbone that enables the invention. So you get these kind of strange effects going on in the field all the time, and it's a wonderful thing as a chemist to watch all of this.
It is a wonderful thing as a human being. It's how progress actually happens, right? And as a person who's done basic research all his career, despite the fact that he wasn't in positions where people think somebody like me would do it, I always understood that at a sort of bone marrow level. And then after you worked in the tRNA company, you also worked in an RNA editing company, right? Which I'm very excited about RNA editing. As for the folks who are listening, all of these technologies, all of these sort of sub-technologies are really derived from what Ionis did initially, Alnylam followed, and so on. And each of them, I think, at different levels and different timings, will have another profound impact on the health of humankind. And so when I think of what's been accomplished, I think of the generations of companies and ideas and so on that have been created out of nothing but a dream. And the only thing in the end that really matters is dreams and whether somebody turns them into reality. And we take it, you and I, for granted that these things all happen, but there was no reason they happened other than the fact that there were people crazy enough to do it, right?
Yeah, that's you're exactly right. And
I'm really crazy. I admit that.
Well, and RNA editing, I think, is a perfect example of that, right? The constructs for RNA editing may be a little bit more complicated. The biologies are not quite as straightforward as RNase H or Argonaut. But the chemistries that were developed at Ionis and Alnylam and others, those have really made the path a little bit more straightforward. Well, a lot more straightforward for companies that are developing new modalities in the space. Things are a little bit more complex because many of those chemistries are kind of plug and play. The 2'-methyls, the methoxyethyls, the phosphorothioate, of course. Phosphorothioate predates even Ionis, of course. But but all of these things come together and make the development of these modalities tractable. Whereas otherwise they wouldn't. And of course, we see these things now even being included in single guide RNAs and possibly even messenger RNAs in the future.
Sure, they will be, and they will need to be if messenger RNAs are to expand beyond vaccine making, in my view. But I do find it amusing when someone says, even an expert like you, that RNA editing is a little more complex than things like RNase H1. In 1989, we didn't know why ASOs caused RNA degradation at all. And there was a big controversy about whether RNase H was actually involved. And so, and having loved that enzyme and I think contributed a lot to understanding RNase H1, I'm very pleased to have somebody think it's very mature and well understood as opposed to RNA editing. It wasn't that way in 1989, let me just say.
Well, yeah, there was so many beautiful papers to elucidate the mechanism from Ionis. I remember a gentleman, Walt Lima.
He worked with Walt Lima and Crook are on all those papers. Walt, John Woo. Yeah, that's all work in my lab.
Right. So, yeah, incredible amount of work, good science went into elucidating those things. So, so it is incorrect of me to imply, I think, that RNase H was somehow simple. Maybe a better way of putting it is that it was just the first enzyme that the people decided to work on, right? To develop into an RNA therapeutic modality.
Yeah, sort of. I think there was only one people who really decided to do it, and that was at Ionis, I think. But anyway, it's very interesting to sit here today and think about the history and how it's all happened. And this podcast series gives me a chance to share some of that with people like you, which is always fun too. I have to be careful and not get too historical because in the end, nobody cares. All they care about is what's next, right? Which is what they ought to care about, right? And Hongene is making what next possible. And that, I think, is the main message we want to give the folks listening to the podcast is Hongene is a part of the creation of the treatment for nanorare patients and a strong supporter of n-Lorem. And certainly we very much appreciate that.
Yeah, absolutely. We're in this for the entire field, and we want to apply our skills, which is RNA manufacturing. We want to do that to the highest quality standards possible, and provide the field with competitively priced raw materials and drugs, and and n-Lorem is definitely high up there on our list of priorities.
Well, again, David, it's great to spend a little time with you, and I hope to see you at the second colloquium this next year. Yeah,
no, that's right. We'll be there. There'll be several of us, I think, from Hongene, including the CEO. The CEO will be there, whom I think you've met, right, Wei? And and that's the end of October, beginning of November, right? So a couple of days. Yeah, we were at the meeting last year. It was a good meeting. So very much looking forward to the meeting this year as well.
Well, we have a lot to talk about. So once again, David, thank you so much for taking the time to do this and for being a strong supporter of n-Lorem. I think our patients and our families need all the help that they can get, and we appreciate all that Hongene has done for us, but really for our patients. Thank you so much.
Thank you very much, Dan. And we're very proud to be supporters of n-Lorem, and that's going to continue into the future. So,
thank you.
Thank you.
n-Lorem is a nonprofit committed to discovering and providing personalized experimental treatments for free, for life, to patients with genetic diseases that affect 1 to 30 patients worldwide, referred to by n-Lorem as nano-rare. Many of these patients progress and die without ever achieving a diagnosis. This is where n-Lorem comes in. They do the impossible by providing hope, and for those that they can help, free lifetime treatment. For more information about n-Lorem or today's episode, visit n-Lorem.org. Any questions can be sent into podcast@n-Lorem.org. Search n-Lorem on Twitter, Instagram, YouTube, LinkedIn, and Facebook to connect with us. This video is hosted by Dr. Stan Crook and produced with the help of the following professionals. Thank you for watching.